Okay, this topic is a general embryology 1. There are two components, general embryology 1 and also general embryology 2. So basically for pregnancy, the normal pregnancy durations are roughly about 40 weeks or 280 days. And the terminology like full term means the birth between week 37 to week 42. Anything birth earlier than week 37 known as preterm. So this preterm birth will increases the risk of complication and premature death because due to underdeveloped lungs, infection due to underdeveloped immune system, problems feeding due to underdeveloped brain, and jaundice from underdeveloped liver, etc. All right, this for preterm birth. The postterm, any birth occurs after week 42. So what are the risks? The risk of complications for both mother and fetus increases significantly. And then the terminology for first trimester from week 1 to week 13, second trimester week 14 to week 27, and third trimester from week 28 to week 41 or 42. I hope you correct your lecture note because I realized just now for third trimester, instead of week 28, I wrote there is a week 38, so please correct it. So what is the parturition? Parturition is the process of giving birth. So this table showing the periods, week in pregnancy, months, the birth, and also classifications. So the period will be fertilizations and also prenatal developments. All right, and then you have first trimester, second trimester, and also third trimester. And what do you mean by embryogenesis? Embryogenesis, and also fetal developments. And normally the embryogenesis will take place about the first 10 weeks or 8 weeks, and followed by fetal developments. Embryogenesis, formation of the embryo. So once the embryo is fully formed, then they will increase in size. It's under the fetal development. All right. So for the first part, will comprises gametogenesis, ovulations, fertilization, and also implantations. There are certain topics in this lecture note already covered in your secondary schools or also the pre-university program. So I will skip on that topics. All right. But before that, let's have a look on the terminologies for gametogenesis. What do you mean by soma? Soma is body tissue. Somatic is a fit to the body. Homologous corresponding in structures. So it means that there are, for example, in male and female, there are homologous structures. It means that they originated or derived from the same structures. What do you mean by germ cells? Cell of an organism which reproduce the spermatozoon for male sex cells and also ovum for female sex cells. So what do you mean by gamete? Gonia is a young sex cells. Site is a adult cells. So this is a chromosome and DNA. I'm sure you're very familiar with these terms, all right? 33 pairs, 46 number, autosome, sex chromosome, XY, and so on. So I will skip these topics. All right. So you can read on your own what do you mean by XX and also XY. So what do you mean by ploidy? Ploidy is the number of copies of each chromosome present in the cell nucleus. So I'm sure you know it's what do you mean by haploid, diploid. So it's a number of copies of each chromosome. So how about N number? N number means number of copies of each DNA molecule in the nucleus. So please do not confuse between ploidy and also N number. All right. Okay. So you can on your own. So please continue. That one's they cover in the secondary schools or pre-university program. Again, type of cell division, there are two types of cell division, mitosis and also meiosis. So mitosis occurs in somatic cells, spermatogonia. Remember I said earlier what do you mean by spermatogonia, oogonia. So this mitochondria and oogonia is a young male and female sex cells. For meiosis, consists of two processes, meiosis phase 1 and also meiosis phase 2. And then for meiosis 1, formation of primary somatocyte and also primary oocyte. Whereas for meiosis 2, production of secondary somatocyte and also secondary oocyte. And phase between the cell division called resting interval. Meiosis 1, between meiosis 1 and meiosis 2. The normal cellular metabolisms, deployed number of chromosomes, 2 and DNA, and mitosis and meiosis has 4 major phases known as prophase, metaphase, anaphase, and telophase. So I'm going to skip all these phases because I'm sure you're very familiar with all these phases. Yeah, the prophase, metaphase, anaphase, telophase, and then for meiosis 1, formation of primary somatocyte, also primary oocyte. So it's a prophase. So this is where the chiasma will occur in prophase. And it's a metaphase 1, anaphase 1, and also telophase 1. All right. Characteristics features of meiosis 1 are what are the main features? Pairing of the matching homologous chromosome. Remember homologous, all right, homologous chromosome. Interchange of chromatic segment between two paired homologous chromosomes. At the end of meiosis 1, each daughter cell has haploid number of double structure chromosome and 2 DNA. All right. For meiosis 2, all right, again, it's a prophase 2, metaphase 2, anaphase 2, and also telophase 2. So during separation of homologous chromosome, segment of chromosome which are going to interchange temporarily remain united. The chromosome structure that has an X appearance known as chiasma. And then purpose of meiosis 1 and meiosis 2 divisions. It will allow interchanging or crossover of a segment of chromatid in prophase 1, creates new chromosome, a mixture from both parents, maternal and also paternal. Provide each germ cells with haploid number of chromosome, half the amount of DNA. Okay, so what I mean by karyotype is a full chromosome set of the nucleus of the cells. Phenotype means entire physical, biochemical, and physiological makeup of an individual as determined both genetically and environmentally. So there's two different definition of karyotype and also phenotype. For gametogenesis, chromosomal and morphological changes of the germ cells will allow for fertilization. So what happened? So basically reductions of chromosome number from diploid to haploid and morphological change to suit the purpose. How? Through mitosis and also meiosis. So basically, what is the differences between gametes, zygote, and so on? So basically, gamete is a new organism produced by fusion of two different sex cells. All right. So sperm united with the ovum and they form gamete. One produced by each parent. These sex cells are called gametes. So this is a definition of the gametes. They are spermatozoa from male from father and secondary side, female gamete from mother. The union when they fertilize and fusion of male and female gamete is called fertilization. The process of fertilization and the cells produce known as zygote. So the first part will be spermatogenesis. Production of sperm occurs in the testis and more specifically in the seminiferous tubule. So it's a cross on the right side, it's a cross section of seminiferous tubule. This where the production of sperm. And it's a cross section of the first tubule. And then, so basically, it contain contain few different types of cells, and namely we have spermatogonium. Spermatogonium is a stem cells. So this mitochondrium will divide themselves or undergo mitosis. Undergo mitosis. And then from there, mitochondrium produce another mitochondrium. Because for mitosis, they're producing two daughter cells. So these two daughter cells, one will remain as a spermatogonium, the other one will develop into spermatocyte. Okay, and it will undergo the meiosis. Yeah. Okay, I'm sorry for the networking problem just now. Where do I stop just now? Which slide? Share. Which slide? Is it this slide or?
The previous slide.
Sorry?
In the previous slide.
In the previous slide. The connection is not that good. Suddenly I'm disconnected. I'm so sorry. Okay, this is a cross section of seminiferous tubule. All right. And then the explanation will be in this slide. So basically in human, it will take about up to 75 days, 64 to 75 days. Begins at spermatogonia, right? Begins in the spermatogonia. So this spermatogonia is a stem cells. They will undergo mitosis. For mitosis, produce two daughter cells. One will remain as a spermatogonium, and the other one will continue with meiosis. Yeah, all right. So the spermatogonia will remain in the basement membrane of the seminiferous tubule. The other daughter cell differentiate into primary spermatocytes, and then the primary spermatocyte will undergo meiosis 1. Then the two cells formed by meiosis 1 are called secondary spermatocyte, and the secondary spermatocytes undergo meiosis 2 with no further replication of DNA occurs. Yeah? And eventually 4 haploid cells resulting from meiosis 2 are called spermatids. So this diagram is showing the process, types of cell divisions. First, the spermatogonia undergo mitosis, will produce two daughter cells, primary spermatocyte, and also spermatogonia. All right. So the primary spermatocyte will undergo meiosis 1, producing secondary spermatocytes, and will further continue with the meiosis 2, producing 4 spermatid. So this spermatid will go maturations known as spermiogenesis. Spermiogenesis is a maturation process of spermatid to become spermatozoa. All right. For male, this spermatogenesis started or begins at puberty. Continues into advanced age, and then, as I mentioned earlier, the sperm production takes about 64 days, 64 to 74 days. Yeah? So what is spermiogenesis? It's a final stage of spermatogenesis. Maturations of haploid spermatid into sperm. No cell division occurs. Each spermatid develops into a single sperm cell. Yeah? All right. Transformation of spermatid into spermatozoa. Formation of acrosome, condensation of the nucleus, formation of neck, middle piece, and also tail, and removing or shedding of moss of cytoplasm. Shedding off moss of cytoplasm. Somatic cells associated with spermatogenesis. What are the other cells? First is the Sertoli cells, very big cells here. Close association with developing sperm. They will support the sperm. They will supply nutrient, and so on, and translocate sperm towards the lumen, and also will control rate of spermatogenesis. And Sertoli cell will secrete about 5% of seminal fluid. Right? So Sertoli cells will support the development of the spermatocyte. The latex cells, also known as interstitial cells, located between the seminiferous tubule, it will secrete hormone. What hormone? Testosterone hormone. A sperm cell consists of three structures highly adapted for reaching and penetrating a secondary oocyte, which is consist of head, neck sphere, midpiece, and also a tail. In the tail, there's a lot of microtubules or structures. In the midpiece, there's a lot of mitochondria. Mitochondria is a power station producing the energy for the movement of the sperm. So the head contains archosomes, and then the enzyme is hyaluronidase and also protease will help in penetrations of the zona pellucida. Nucleus with haploid number of chromosomes. Right? The midpiece, many mitochondria provide ATP in locomotion, is energy. And the tail is a typical flagellum, propel the sperm cell along its way. For oogenesis, right? Types of cell division for oogenesis, right? Before birth, the cell known as the oogonia, and then after birth, all the oogonia will develop into primary oocyte. At birth, each ovary contains about 250,000 or 250 primary oocyte, and they are arrested at prophase meiosis 1. Right? So as compared to male, the spermatogenesis started at puberty. But for female, it started before birth. Started with oogonia, and just before birth, all the oogonia will change into primary oocyte. And then we will arrested at prophase of meiosis 1. At puberty, completion of meiosis 1, producing 2 daughter cells. Yeah, right. One of them is a polar body, and also secondary oocyte ovulated in metaphase of meiosis 2, and they will undergo begin the tertiary follicle and complete only if fertilization occurs. Yeah, under meiosis 2 and producing 4 daughter cells. Yeah? If fertilization occurs after ovulation, meiosis 2 is completed, or will become mature site. Right, this is for the oogonia. I have another diagram which is more comprehensive. So this diagram showing the prenatal before birth and also postnatal maturation after birth. Okay. Up to 7 months of fetal development, all the oogonia will undergo mitosis and form the primary oocyte. Just before birth, there is no more oogonia. All they are primary oocyte. Right? And this primary oocyte will undergo a meiosis 1. Meiosis 1, but will be arrested at prophase 1. They remain dormant, or we call it a diploid stage or resting stage. This resting because of the inhibitor known as oocyte maturation inhibitor secreted by follicular cells. Yeah? This is before birth. Okay. At birth, each ovary contains up to 1/4 to 1 million of primary oocyte arrested at prophase meiosis 1. Majority will be atretic. Majority will be atretic. Atretic means disappear, die, and so on. So the total, and then beginning of puberty, roughly the remaining about 400,000. That when this person reaching at puberty, when reach the puberty around 11 to 30 years old, all right? So that's why the starting of the ovarian cycles. So that's why the start point of ovarian cycles. So each ovarian cycles, roughly about 5 to 15 of the primary oocyte will begins to mature. So not all, only about 5 to 15 will resume, begins to mature. However, usually only one reach fully maturity. Others will degenerate and also become atretic. So each ovarian cycles up to 15 of primary oocyte will continue development, but only one will reach full maturity for each ovarian cycle. So they will continue. The first meiotic division resume shortly before ovulation. Shortly before ovulation. And they are producing the secondary oocyte. All right. And then just before the ovulation, it will continue with the meiosis 2. Meiosis 2, but they are arrested at metaphase 2. Arrested at metaphase 2, unless if there is a fertilizations, so this meiosis 2 will continue and completed. All right. So basically, fewer than 500 oocytes will be ovulated in the reproductive lifetime of the individual. No fertilizations, oocyte will degenerate 24 hours after ovulation. So this diagram is showing the prenatal maturation and also postnatal maturation. So as compared for male, the process is a continuous from meiosis 1, meiosis 2, but for female, they are arrested at different level. All right. The first will be in the P1, second in the metaphase 2. So these are primordial germ cells, all right, detectable at week 4 of development in human, migrate from allantois to genital region. Called these primordial germ cells. And then these primordial germ cells are covered by primordial follicle, single layer follicle cells. All right, single layer follicle cells in the primary oocyte, arrested in prophase 1, significant oocyte differentiations, and also present at birth. So these primary follicles at puberty, develop at puberty, oocyte will complete growth, forming the zona pellucida. And then the primary follicles, multiple layer follicle cells, they are stimulated by follicular stimulating hormone, FSH. And these are Graafian follicles. These are follicles, the indication will be fluid filled and there's a cavity here. And then the oocyte is a secondary oocyte, right? So this is a process developments of the primordial germ cells, okay, primordial follicles, primary unilateral follicles, primary multi-laminar follicles, Graafian follicles, and then ovulations. And then the remaining follicles here will develop into corpus luteum. Right? If there is no pregnancy, corpus luteum will degenerate and form corpus albicans. If there is pregnancy, corpus luteum will remain for a few months to secrete hormone, to sustain the pregnancy. So this different stages. You can see this from the ovary. So you can see here, there's a primary oocyte. There's a primary oocyte. It's the Graafian follicles, primary follicles, and so on. Again, different stages. Primordial follicles with primary oocyte, and this is maturing follicles, formation of zona pellucida, and then the follicle cells here forming the granulosa cells. Yeah, the follicle will form the granulosa cells, and then the surrounding here, they form a theca, theca interna and also theca externa. That's another layer. Okay, ovulations. Ovulations. This Guyvan follicle increases in size. The diameter is about not 15 millimeter. It is 1.5 millimeter, and then stimulated, influenced by FSH and also LH, luteinizing hormone. Surface of ovary bulge locally, will bulge locally, all right, and they are vascular, known as stigma. So it's where the oocyte will be excreted. The local weakening and degeneration of ovarian surface, increasing intrafollicular pressure, muscular contraction of the ovarian wall. Those contractions will cause extrusion the oocyte. Oocyte will be extruded. Okay. And then the oocyte together with the cumulus oophoris, and initially these cumulus oophoris, they are granulosa cells. Once they are ovulated, this granulosa known as cumulus oophoris. Cumulus oophoris. Arrange themselves around the zona pellucida to form corona radiata. Okay, corona radiata. So this cumulus oophoris arrange themselves around the zona pellucida to form corona radiata. And then the remaining granulosa cells and also the theca interna are vascularized. Under the influence of luteinizing hormone, self-developed yellowish to form a luteal cells and eventually form the corpus luteum. From the corpus luteum, they will secrete progesterone. Stimulation of progesterone and estrogen causes the uterine mucus to enter progestational or secretory stage in preparation for implantation of the embryo. So, how about the oocyte transport? Once the oocyte excreted, fimbriae, the terminal end of the uterine tube, fimbriae is they contain a finger-like projection. Yeah? So they will sweep over the surface of the ovary. Yeah? So this finger-like projection, known as fimbriae, will sweep over the surface of the ovary, and the tube will contract rhythmically. Right? Oocyte is carried into the tubes by sweeping movement of fimbriae and also motion of cilia, on the epithelial lining, and then we reach the uterine lumen in 3 to 4 days. Corpus albicans, if fertilizations fail to occur, corpus luteum reach maximum development about 9 days after ovulations. Subsequently decrease in size through degeneration, become fibrotic scar tissue known as corpus albicans. Yeah? If there is no fertilization occurs, roughly after 9 days, this corpus luteum will degenerate, forming the corpus albicans, generating corpus albicans. If there is a pregnancy, the corpus luteum will remain and producing the hormone progesterone. So this is a correlate, it's a clinical, it's a plaque anatomy, menstrual pain, known as erythematous pain, increased basal body temperature during ovulation, failure to ovulate due to diminished concentration of gonadotropins. Yeah? So if oocyte is fertilized, degeneration of the corpus luteum is prevented by human chorionic gonadotropin, HCG. So corpus luteum continue to grow and form the corpus luteum of pregnancy. Yeah? And then they only support for the first few months only. Yeah? Okay, fertilizations. The process by which male and female gametes are fused, they occurs in the ampullary region of the uterine tube, the ampulla. of the uterine tube, and the sperm will undergo capacitation and also acrosome migration. Yeah. What is the capacitation? It's a period of conditioning in female reproductive tracts. So about 7 hours, where the glycoprotein coat and seminal plasma protein are removed from plasma membrane. And this acrosome reaction occurs after binding to the zona pellucida, induced by zona proteins, result in release of enzyme to penetrate zona. Okay, phases of fertilization, there are 3 phases, phase 1, phase 2, and so phase 3. Phase 1, this is where the penetration of corona radiata. Phase 2, penetration of zona pellucida. And phase 3 is a fusion of oocyte and sperm cell membrane. For phase 1, penetration of corona radiata, right? So this is a corona radiata from the granulocyte cells. Of the about 2 to 100 million spermatozoa deposited in the female genital tract, roughly about 200 to 300 reach the site of fertilization. Yeah? Only one of these fertilized the egg. The others ate the fertilizing sperm in penetrating the pairs, protecting the female gamete. And only the capitated sperm pass freely through corona cells, or corona radiata. So phase 2, penetration of the zona pellucida. This is zona pellucida. Once head of the first sperm comes in contact with the membrane of oocyte, permeability of zona pellucida will change. This phenomenon known as zona reactions. So this zona reaction will prevent penetration of other sperm. So the first sperm that in contact with the zona pellucida, it will cause zona reaction. So this zona reaction will change the permeability of the zona pellucida. It will prevent penetration by other sperm. So the other sperm, they are embedded in the zona pellucida. Phase 3, fusion of oocyte and sperm cell membrane. As soon as the spermatozoa come in contact with the oocyte cell membrane, the two membranes fuse. Yeah? As soon as the spermatozoa come in contact with the oocyte cell membrane, right, the two membranes will fuse. In human, both head and tail of the spermatozoa enter the cytoplasm of the oocyte. Membrane of the spermatozoa does not enter. Will remain in the site. So the oocyte response, as soon as the spermatozoa has entered the oocyte responds in 2 different ways. Cortical and zonal reaction, resumption of meiotic II cell division, and also metabolic activation of the eggs. So first, cortical and zonal reactions, 2 reactions take place. First, as I mentioned earlier, oocyte membrane become impermeable to other spermatozoa. Second, zona pellucida change structure, prevent endosperm binding and penetrations. Second, resumption of meiotic II cell divisions. Right? So basically, immediately after the spermatozoa enter the oocyte, oocyte is undergo metaphase 2. The oocyte completed second meiotic division, and 2 daughter cells are formed. All right. One of the daughter cells receive almost all the cytoplasm known as definite oocyte. The other daughter cell hardly receive any cytoplasm known as a second polar body. So 22 plus X chromosome of definite oocyte arranged in the nucleus known as female pronucleus. So the metabolic activation of the egg, the activating factor is from the spermatozoa. Remember, the activating factor for metabolic activation of the egg came from the spermatozoa. All right. Immediately after ovulation, oocyte shows spindle formation of the second meiotic division. Okay. Spermatozoa move to the female pronucleus, and then nucleus spermatozoa becomes swollen and form male pronucleus, while the tail degenerates. There are two pronucleus, male and female pronucleus. And then DNA of each pronucleus will replicate. And 23 maternal plus 23 paternal chromosome arranged on the spindle, split longitudinally at the center. Each split chromatic moves to the opposite pole to complete the first mitodivision after fertilization, forming two-stage cells. Will undergo mitotic. So these two will further mitosis. So it will double in number, 2, 4, 8, 16, 32, and so on. So this two cell stage of zygote with normal diploid number of chromosome is formed. So what are the main results of the fertilizations? The first will be restoration of the diploid number of chromosome, and then determination of a sex factor. And then the sex factor actually is determined by the paternal factors. So remember, the gender of the fetus of the baby is determined from the paternal factors and not the mother factors, because the mother only carries the X chromosome, whereas for Y chromosome is from the father factors. So don't blame the mother, but blame the father if you have more female than male children. So normally people will blame the mother because of having more female, more girls other than the boys. So that's a paternal factor, not the maternal factors. All right. So I don't know why they blame me. Okay. And then initiation of cleavage, fertilized ovum start dividing by mitosis cell divisions. So it's a clinical correlation, applied anatomy, never mind, and skip. So once they forming the zygote, the process of mitosis cell division of the fertilized ovum, and then two cell stage zygote surrounded by zona pellucida undergo cleavage and produce more number of cells. Cells become smaller with each cell divisions. Zygote now known as blastomeres. So you have zygote, we have single cells divided into two, and then from two we become 4, 4 become 8, and so on. So this individual cell, we call it blastomeres. If this blastomere separated, for example, as two-stage cell, if this blastomere separated, so you will have a twin. You will have a twin. So this is showing the 2-cell stage, 4-cell stage, and then the 2, 4, 8, 16, and 32. 32 normally they form a morula. It's a packed cells known as a morula. So approximately about 3 days after fertilization of the cell divide again to form a 60 cells, and that's where the starting, we call it a morula or mulberry, and from 60 become 32. 4 days after fertilization, morula enter the cavity of uterus and zona pellucida will disappear. And then this morula cells, it's a number of cells. So these cells will group themselves into inner cell mass and also outer cell mass. Okay. So inner cell mass and also outer cell mass. The inner cell mass will give rise to the tissue of the embryo proper, while the outer cell mass form the trophoblast, which later contribute to the formation of the placenta. Yes, let me say, okay. So basically here, initially from the morula here, so these cells here, cells will group themselves, the inner cell mass and also the outer cell mass. The inner cell mass will develop into embryo proper, and then the outer will become trophoblast and will contribute to the formation of the placenta. Blastocyst formations. So remember just now the morula, you have inner cell mass and also outer cell mass. Once cavity is formed in the morula, First, the cell group themselves, inner and outer, and then the further development will be formation of a cavity. So this cavity known as blastocyst cavity. So once the blastocyst cavity appear, then morula will become blastocyst. Okay. Remember the zygote, blastomeres, morula, and now blastocyst. Right? Fluid penetrate into the morula and a single space known as blastocyst or blastocyst cavity is formed. Morula with blastocyst or blastocyst cavities known as blastocyst. Right? Blastocyst cavity push a group of cell inside the inner cell mass to one pole. So basically, remember the stages just now. So you start with the zygote, blastomeres, morula, and now blastocyst. So in embryology, this is a process of development. When they change their shape or function or structure, then we give them a different name.
Doctor, I have a question.
Yes, please.
Where does the fluid that penetrates into the morula came from?
Sorry, fluid?
Where does the fluid that penetrates into the morula came from?
Surrounding the tissues there. Because there's a lot of fluid surrounding the we call it the morula around them. Because they are still in the. Okay, just remember the fertilization occurs in ampulla, right? In uterine tube. Of course, the uterine tube itself, there's a lot of epithelium cells. So this epithelium cell also will, we call it, they will secrete nutrient, everything, and of course they will secrete fluid as well. All right. And then bear in mind, remember the cell, the initial cell, the blastomere. All right. So because they are reducing in size, so they also contain water as well, the fluid. All right. We are talking very minute amount of fluid. Okay. So from the surrounding and also from the blastome itself. All right. Okay.
Thank you, doctor.
All right. And then the inner cell mass now called the embryoblast, which later developed to form the tissue of the embryo. All right. The covering cells, outer cell mass, form the trophoblast, which later contribute to form a placenta. So this is a diagram here. So this diagram here. So basically, the initially is your outer cell mass. Outer cell mass eventually they form trophoblast. Trophoblast, and then initially the inner space here full with inner cell mass, right? Inner cell mass. Okay. So this inner cell mass, suddenly there's a penetration of fluid, accumulation of fluid. Once there's a accumulation of fluid, they will form a cavity. This cavity is known as blastocyst cavity. And because of this cavity, the inner cell mass will push to one side or to one pole. And now we call it embryoblast. So embryoblast derived from inner cell mass. Right? Inner cell mass. So the whole thing here, they are blastocysts. Blastocysts. Right? So the blue color, they are embryoblast. Embryoblast will, in future, we develop into embryo. All right. And this where the outer cell mass here, the trophoblast, the top of cells. After that, it will start invading the uterine wall for the implantations to be implanted in the uterine wall. So these trophoblast cells, okay, they will start to invading the uterine wall. Okay. Remember, embryology is something that you need to imagine. You need a good imagination. So the implantations, 6 days after fertilizations, trophoblast cell over the embryoblast pole penetrate the mucosa of the uterus. By the end of first week after fertilization, zygote is implanted in the mucus of the uterus, and this process known as implantation. So implantation. At the time of implantation, mucosa of the uterus is in the secretory phase. Secretory phase. So look here. That's how the process, starting with the ovulations, and then transported to the uterine tube, and then fertilized in the ampulla region, and then from here will be transported to the uterine wall, So the fertilized ovum will undergo developments, cell divisions from single to double, 2-cell, 4-cell, 8-cell, 16 cell, 32 cells, known as morula, And from here, from morula, there's a formation of a cavity, blastocyst cavity. Then this become blastocyst. Then you have outer cellma and also inner cellma. The inner cellma will become embryoblast. The outer cellmas will become trophoblast. And this trophoblast will start invading the mucosa, the uterine mucosa. And then, so this mucosa at the state of progesterone stage, and then it will be implanted in the wall here, in the uterine wall. All right. So basically, this is how the phases of menstrual cycles. All right. It started with the bleeding on day 1. Started with the bleeding, and then the mucosa will undergo phases. All right. For example, follicular or proliferative phase, progestational and also secretory phase. And when they are fully implanted, it's a gravid phase, the thickening of the mucosa. If there is no implantations, then the process will start all over again, bleeding and so on. Right? Okay, every ovarian cycle. Right? If you have any question, you just simply interrupt me now. There's no problem. Right? Just ask. Okay, the second part, the general biology 2, I'm covering the following topics: bilaminar jaundice, trilaminar jaundice, followed by embryonic period and also teratology. All right. Bi means two layers. Trilaminar means tri layer. Embryonic period means when they become embryo. So this terminology followed by cover, line, vaginate, renal, caudal, diverticulum, edema, and edematous. Followed by villus, fetus, fetal, decidua, embryonic pole, or embryonic pole. All right, you can go on your own. All right. So as I mentioned earlier, for cleavage, some mitosis will become a lot of blastomeres, and then following the third cleavage, blastomere maximize their contact, become compactions and form the morula. Right? And then some would say 16 cell state, some would say on the 32 cell state, doesn't matter. Right? And then there's the inner cellma and also outer cellmas. Right? Just to recap, 2 cell state, 4 cell state, and also morula. So these are individual blastomeres. Okay. All right, a different diagram. All right. So this is a blastocyst formation I mentioned earlier. All right. Cross formation of the cavity, blastocele. All right. And then the trophoblastic cells will start penetrate the uterine mucosa as early as day 6. Again, formation of the blastocyst when there's a blastocyst cavity. And then there's an inner cell mass. So this inner cell mass will develop into embryoblast. Again, it's a blastocyst. It's a blastocyst. Trophoblast is here. And this is your embryoblast or inner cell mass. Again, in a different diagram, I already showed on the earlier lecture. All right. So these are the process when they start invading the endometrium wall or the mucosa layer. Right? This is all the process already mentioned earlier. All right. So starting with the ovulations, collected by the fimbriae, fertilization in the ampulla, and then transported into the endometrium. Right? And the process in the zygote itself. And then, okay, first bilaminar germ disc. Okay, now day 8, blastocyst partially embedded in the endometrial wall or stroma. All right. So this trophoblast, remember the trophoblast, the outer cell mass here. So this trophoblast will develop into two cytotrophoblast and also syncytotrophoblast. Yeah? So basically, the cytotrophoblast, it will remain covering the embryoblast here. All right. And then the syncytotrophoblast, the one who invading the endometrium wall. So here's your syncytotrophoblast, here's your cytotrophoblast. It's a cytotrophoblast. All right. So this embryoblast, remember the embryoblast here, will develop into two layers known as hypoblast and epiblast. So there are two layers. That's why it's a bilaminar germ disc. So it is a germ disc now. It's a germ disc. Bi means two. Bilaminar. So bilaminar consists of hypoblast, the ventral part here, and also epiblast, bilaminar germ disc. And then there's a group of cells in the embryoblast. A group of cells in the embryoblast will forming a cavity, known as amniotic cavity. Okay, amniotic cavity. So if you look back here, this diagram, so it is a inner cell mass developed into embryoblast. So this embryoblast will form bilaminar germ disc, which is epiblast and hypoblast. Yeah? And then there's a group of cells here will form amnioblast. Amnioblast that eventually will form a cavity known as an amniotic cavity, and this amniotic cavity will fill with amniotic fluid. I'm sure you heard before amniotic fluid, right? So this is amniotic fluid. Okay. And then the endometrial stroma become edematous and highly vascular to support the developing fetus here. All right. Okay. So again, this blastocyst will almost fully embedded in the endometrium wall. So again, by looking at this diagram, there are a few such that you can see. The trophoblast divided into two cytotrophoblasts. Right? Okay, that's still covering the blastocyst, the cytotrophoblast, covering the blastocyst, and then the other one is a syncytotrophoblast, the one invading the endometrium wall. Okay, so again, syncytotrophoblast. So this amniotic cells producing the amniotic cavity. This amniotic cavity will fill with amniotic fluid. So it's a bilaminar cavity, bilaminar germ disc, epiblast and hypoblast. And then remember, this is a blastocyst cavity. So this blastocyst cavity will transform themselves or change themselves into exocoelomic cavity. So remember, they changed their name. So this blastocyst cavity will become exocoelomic cavity. In the exam, I might ask you, for example, exocoelomic cavity derived from something like that, blastocyst cavity. All right. And then the outer layer is your cytotrophoblast. Actually, the name change when there's a formation of exocoelomic membrane. There's a formation of exocoelomic membrane covering the blastocyst cavity that it changed their name. Okay, become exocoelomic cavity. So this exocoelomic membrane known as user membrane as well. HEUSER, user membrane. All right. So as for now, okay, and then they are fully embedded in the endometrium wall, and then here's your amniotic cavity with amniotic fluid, and here's your cytotrophoblast. This is a syncytotrophoblast. This is an endometrium wall, maternal endometrium wall. There's a lot of blood vessels, right? So when they erode the endometrial wall, when the syncytotrophoblast eroding or invading the endometrial wall, so they are invading or eroding the blood vessels of the maternal. That's where the developing zygote here will receive nutrients from the maternal once they erode or when they're reaching the maternal blood artery, the arterial system of the placenta later on. All right. So it's still in the endometrial wall. Okay, day 9, blastocyst more deeply embedded. And then there's a vacuoles in syncytotrophoblast. Okay, here's your syncytotrophoblast. They form a vacuole known as a lacuna. And this known as a lacuna stage. The flat cell from hypoblast, will form exocoelomic membrane, as I mentioned earlier, exocoelomic membrane, all right, known as user membrane. The same thing, user membrane. It will line the exocoelomic cavity, line the blastocyst cavity. Once they line the blastocyst cavity, change their name. The new name will be exocoelomic cavity, or also known as a primitive yolk sac. So primitive yolk sacs is a exocoelomic cavity. Okay, exocoelomic cavity. Day 11 to 12, fully embedded. Okay, and then remember just now the syncytotrophoblast eroding the endometrial wall, it will erode the blood vessel as well. Okay, and then the lacuna space form the intercommunicating network, known as the embryonic pole. All right. And then the syncytotrophoblast penetrate deeper and erode the maternal capillaries. And then once they erode the capillaries, the lacuna fill with maternal blood. That's where the starting point of uteroplacental circulations. If in the exam, when uteroplacental circulation started, it started when the maternal capillaries enter the lacuna of the syncytotrophoblast. How they can penetrate, how they can enter, how the blood of the maternal enter the lacuna? Because of the syncytotrophoblast penetrate and erode the maternal capillaries. All right, that's the starting point of the uteroplacental circulation. Uteroplacental, okay, from the uterus to placenta, because in this area here, it will develop into the future placenta. Okay, future placenta. That's where the developing zygote here will receive nutrients. Bear in mind, the developing zygote will not receive direct blood from the maternal. They will not receive a direct blood, but it will transport the nutrient and will remove the waste product. So remember, there is no direct blood circulation from maternal to developing fetus. Cell derived from yolk sac cells, known as extra embryonic mesoderm. Remember the yolk sac just now? Okay. Okay. Cell derived from the yolk cell will derive into extra embryonic mesoderm in this area. They become thick, and form the extra embryonic mesoderm. All right. Okay. Because this extra embryonic mesoderm, okay, eventually in future, they will form a cavity. It will form a cavity. So that cavity will form the new cavity known as chorionic cavity in here. It's an extra embryonic xylem. It's a chorionic cavity. All right. And then because of this mesoderm forming cavity in the middle, of course, there are two mesoderm membrane, known as somatopleuric and also splenopleuric. Okay. So the splenopleuric that covering the the yolk sac, all right, and then the somatopleuric will attach to the trophoblastic layer here, cytotrophoblast. This is a somatopleuric mesoderm. I will show a different diagram here. All right, so at this stage, can you see here the extra embryonic mesoderms? All right. So this extra embryonic mesoderm will become thick and will form a cavity. Will form a cavity. Then eventually this cavity unite and form a chorionic cavity. And then cells of the endometrium loaded with glycogen and lipids, intercellular space filled with extravasate, tissue become edematous, and this condition known as a decidual reaction. This scenario known as a decidual reactions. So the extra embryonic xylem expand will become chorionic cavity. Remember just now this show you this small space here. So this is small cavity here. The coelomic cavity will unite and become a bigger cavity. So this bigger cavity known as a chorionic cavity. Okay, chorionic cavity. Right. So now, how many cavities are there? Of course, there is a amniotic cavity. Then you have a yolk sac, and now it's a chorionic cavity. There are three cavities at this stage. Right? Remember? Okay, amniotic cavity, yolk sac cavity, and also chorionic cavity or coelomic cavity. Okay, coelomic cavity. So this sometimes they this coelomic cavity is sometimes they call it a chorionic cavity. We are referring to the same structure, right? And then the extra embryonic mesoderm lining the inside of cytotrophoblast known from the chorionic plate. Yeah? Okay, chorionic plate, or sometimes called chorion. And then forming the precordial plate. That's okay. That's all right. Yeah? Okay, the bigger pictures here. All right, the three cavities: amniotic, yolk sac, and also chorionic or coelomic cavity. Okay. So here's your precordial plate. Never mind, it's not that clear. Okay. On day 13, surface defect will heal. And then if there's a bleeding, implantation bleeding increase blood flow into the lacuna. So now, if you look here carefully, the diagram on the right side here, let's see carefully, all right? This chorionic cavity become bigger and bigger. And then this cavity will push the tissue surrounding this blastocyst, all right? And they are connected to the developing placenta through a connecting stalk. Right? Through a connecting stalk. And this connecting stalk in future it will elongated and form umbilical cord in future, right? Become, okay. And then these are developing placenta. The lacuna is filled with maternal blood. And then on day 30, the trophoblast, remember the trophoblast layer here, to form a primary villi. This is a process of formations of the placenta. Right? And then the hypoblast cell migrate along the inside of the exocellular membrane and will become a secondary yolk sac. This is an abnormal implantation. Give me 1 second. Okay, how about the abnormal implantation site? It can be extrauterine, means outside the uterine wall, or we call it an ectopic pregnancy. So this is a normal implantation will be in the wall of uterine wall, but the ectopic will be other than that. So it can be in various locations. It can be along the uterine tube, or it can be in the internal os, or it can be in the cervical implantations, or it can be in the ovary itself, or it can be in the mesentery on the intestine wall. Right? So there's an ectopic pregnancy. So it can be in the internal os of uterus, in the ovary, in the femur, in the uterine tube, in the mesentery, and so this is an ectopic pregnancy. And again, showing the ectopic pregnancy where they develop in the rectouterine pouch instead in the uterus. Okay, now it's a trilaminar jaundice. We're done with the bilaminar. Bilaminar is a epiblast and hypoblast. So it's an epiblast, hypoblast, amniotic cavity. It's a yolk sacs, and it's a chorionic cavity. So that is a chorionic cavity. So when the bilaminar become trilaminar, from 2 layers become 3 layers, known as trilaminar jaundice. The process known as a gastrulation. So gastrulation, the process that establishes the 3 germ layers in embryo. So the trilaminar germ layer, because there are 3, the 2 layers known as a ectoderm, the outermost layer, ectoderm, mesoderm, and endoderm. So basically here, so here's your epiblast, and also hypoblast. So the theory is that a group of cells in the epiblast, will migrate medially, and invaginate inferiorly, all right, forming additional 2 layers. All right, so the epiblast will become ectoderm. The epiblast will become ectoderm. The new layer will be mesoderm in the middle, and the endoderm, the most ventral part. All right, and the hypoblast will be displaced, right, by the endoderm. The primitive streak on the surface of epiblast. All right, so here's your primitive streak, and then on the cephalic end is a primitive node. Okay, and then the cardiogenic area or heart forming plate. Okay, now let's see some pictures. Okay, now this from the superolateral view. All right, and above blue color here is a cavity. What cavity are there? They are amniotic cavity. Okay, amniotic cavity, and then you have primitive streak. What does a streak look like actually? Okay, if you go to the beach and then there's a sand, right? Just use your finger, and then on the sand, then you move your finger on the surface of sand. That's how the streak looks like. Okay, so you on the sand and you move your finger. That's how the streak. Not here is a primitive streak. And then on the cephalic area here is the area of the precaudal plate. Precaudal plate. And at this level is bilaminar, epiplast, and hypoplast. So the gastrulations, okay, the process formation of the trilaminar gymnast, the cells from the epiplast will migrate toward the primitive streak, will migrate medially and vaginate inferiorly, and forming additional 2 layers, mesoderm and endoderm. So here, the arrow shows that the cells will migrate from the epiblast, will migrate medially, and then vaginate inferiorly, and will form additional layer, 2 additional layers, mesoderm and endoderm. All right. This from the superior view, and this area of precaudal plate actually in future will develop into your mouth area, whereas the glaucal membrane will develop into your urogenital system, the opening of the urogenital system. All right. So these are the cephalic part towards the head, this is towards the tail, right? From the superior view. Now, formation of the 3 layers, okay? Ectoderm, mesoderm, and endoderm. From where? From the epiblast. Okay. There's another structure, formation of the notochord, the one in the center here. Now, the pre-notochordal cells invaginate in the primitive pit, move forward in cephalic directions. Okay, if you look here, this is your primitive streak. There's a primitive node. In the middle is a pit, primitive pit. There's a group of cells from the epiblast to form as a notochordal cells. Notochordal segment in future we develop into notochord. So these notochordal cells will invaginate in the primitive pit and will migrate cephalically and will form a notochord. Okay. So here, from the primitive pit here, will migrate cephalically towards the head, forming the notochord. Okay. So it's a notochordal plate here. So eventually, they will form a notochord. Notochord in the middle. Okay, notochord in the middle. All right. So from plate, it will detach from endoderm and form the definitive notochord. All right. This diagram showing how the notochord will form. So what is the notochord actually? Notochord is define the primordial axis of the embryo, and it will give it some rigidity, some sort of a support to the developing embryo. Serve as a basis for the development of the axial skeleton. That's where the exoskeleton will develop. All right. So remember, from the superior view, here is the primitive streak, and this is your notochord. Notochord will develop cephalically. Okay, cephalically, and then the embryo will become longer and longer. Right. So we'll give it some rigidity, serve as the basis for the development of the axial skeleton. Axial skeleton. It indicates the future side of the vertebral body. Okay. Indicate the future side of the vertebral bodies, and also will induce overlying ectoderm to form a neural plate. So now, this is a cross section. Cross section here. This is a notochord. Ectoderm, the blue color, red color is your mesoderm, and the yellow is a endoderm. So this notochord will indicate the future side of the vertebral body, your backbone, your backbone. And then it will stimulate or induce the ectoderm around here to form a neural plate. So this neural plate eventually become a neural tube. So this neural tube eventually will develop into your spinal cord and also your brain. So remember, in adults, they remember the vertebral column, the bone, and then inside there is a spinal cord. Okay, and then will connected to the brain. So basically, the brain and spinal cord, they develop from the neural tube. So this neural tube developed from neural plate. So this neural plate developed from the midline part of the ectoderm. Okay. So the glaucal membrane formed at the caudal end of the membrane dish. Okay. And then the allantoenteric diverticulum here will form the allantoids on the day 16. Okay, growth of the germ disc. Growth of germ disc is superior view. Superior view is a primitive node, primitive streak. All right. Initially flat and almost round. Initially flat and almost round. Gradually become elongated with a broad cephalic, and a narrow caudal end. Okay, broad cephalic end and narrow caudal end. Imagination of surface cells in primitive streak continue until end of week 4, then regresses and diminishes in size. Okay. So gastrulation is a highly sensitive stage. Gastrulation is the formation of trilamina germ disc. High dose of alcohol will kill cells in the anterior midline of the germ disc, and then deficiency of the midline in craniofacial structures. It can cause deficiency of the midline in craniofacial structures, and also will cause hollow prosencephaly, right? Or it can cause caudal degenesis, failure of the caudal part to form, right? Due to insufficient mesoderm formed in the caudal moss region. Abnormalities in lower limb, urogenital system, lumbosacral vertebrae, and also sometimes this condition due to maternal diabetes, if not controlled. And this is sacrococcygeal teratoma, remnants of primitive streak. Remember the primitive streak just now? So it's a remnant of primitive streak, most common in the form of tumors in newborn. All right. Okay. Future further developments of the trophoblasts is where they will develop into the placenta. So remember the villi just now, okay, the primary villi and also secondary villi. The first villi is a primary, and then the second will be your secondary villi. End of third week, there is a tertiary villi, definite with placental villus. The outer cytotrophoblast shells attach the chorionic sac firmly to the maternal endometrium. So here again, this diagram. So the placenta will further develop or take place on this part only. The remaining here will become become shrunken because the placenta will be on this area. Okay. So this never mind anchoring villi, what by terminal villi. Okay. What's the embryonal period? Embryonal period is from week 3 to week 8 of development. It is a period of organogenesis. Organogenesis is where the formation or starting point of the formation of the organ. Okay, so each germ layer gives rise to specific tissue organs. Remember the 3 layers: ectoderm, mesoderm, endoderm. It will give rise to specific structures. Yeah. So main organ system established by the end of the period. Major features of external body form recognizable by end of second month. Yeah. Okay, first, the ectodermal. The ectodermal. Remember the ectodermal? Okay, it's your ectoderm. It's your notochord. Okay, beginning to week 3, formation of the neurulations. Neurulation means a process. It is a process formation of the neural tube. Neural tube. This neural tube eventually will develop into your spinal cord and also brain. Okay, so this neural groove will unite, eventually become a neural tube. Okay. So at the same time, there's a group of cells here in the ectoderm here. Sorry, in the ectoderm, known as neural crest cells. Yeah? So this the yellow color here is the neural crest cells. So these neural crest cells will contribute to the following structures. Yeah? Will contribute to the formation of the following structures. What are the structures derived from neural crest cells? They are all the ganglion, Schwann cells, meninges, medulla of adrenal gland, melanocyte, connective tissues of craniofacial structures, and also part of the heart contributed or derived from neural crest cells. Yeah? Again, initially they're almost round. The cephalic will be broader, the caudal will be narrower. Now, the ectodermal thickenings in cephalic region, okay, in cephalic region, and formation of the optic placodes and also lens placodes. Optic placode means in future we develop into the eye, your eye area, yeah? Your eyeball. Okay. Ectodermal germ layer gives rise to organ and structures that maintain contact with the external environment. So what are the structures derived from the ectoderm? They are central nervous system, peripheral nervous system, sensory epithelium of ear, nose, eyes, your epidermis, subcutaneous gland, mammary gland, pituitary gland, and also enamel of teeth. In the exam, for example, the question will be, the following structures are derivative of the ectodermal germ layer, except, or in the SAQ, I can ask you, name 3 derivative of ectoderm germ layer, something like that. It can be mesoderm or endoderm or ectoderm, right? That's how the question will be asked. All right. How about the mesoderm germ layer? In the red color here. Form a thin sheet of loosely woven tissue. By day 17, cell close to midline proliferate. Proliferate will thick to form paraxial mesoderm. Paraxial mesoderm. Laterally is a lateral plate mesoderm. So basically, the mesoderm will divide into 3 parts. Most middle will be paraxial mesoderm. The most lateral will be lateral plate, and then in between is a intermediate mesoderm. Sorry, intermediate mesoderm. All right. Okay. This is the intermediate mesoderm here. If you look carefully, it's a paraxial. It's a paraxial, and it's a lateral plate, and in between is the intermediate mesoderm. And then beginning of the 3rd week, paraxial mesoderm organized into segment known as a somatomes. Somatomes. It started in somite, all the somite here. All right. It started in the cephalic region. Right. From occipital region, the somite first pair appears on day 20, and subsequently 3 pairs per day will start appear. All right. By the end of week 5, roughly about 42 to 44 pairs. Right? So this all the somites, right? The number of somites. So age of embryo is expressed in number of somites. So because the first somite appears normally on the day 20, by counting the number of somites, then we can postulate the age of the embryo. Okay. So this somite will further develop into what? Okay. So this is a somite here. All right. And then this somite will develop into sclerotome, myotome, and also dermatome. Okay, remember your mesoderm, your paraxial, intermediate, and lateral plate mesoderm. This paraxial will develop into somite. So this somite will further develop into three parts: sclerotome, myotome, and dermatome. By looking at the name, myo means muscles. Derma means your derm is your skin. Okay. Your derm is also subcutaneous tissues. Yeah. So sclerotome is a vertebral column, your backbone. Your backbone derived from sclerotome. All right. And then dermomyotome will divide into two. Myotome and also the dermatome. Right? Dermatome is a dermis and subcutaneous tissues. Yeah. The intermediate mesoderms, I'm just giving the gist or the main structures, and not into detail because it's only 3 hours lecture, so we cannot go through all the process. Just sufficient for you to appreciate what structure derived from which layer of germ disc. Right? For intermediate mesoderm, cervical and upper thoracic region form segmentally arranged cell cluster, right? And they form the nephrotome as well. Nephrotome, something got to do with the nephron. Right? So unsegmented mesh tissue, nephrogenic cord, it will develop into excretory unit of the urinary system and also the gonads. Yeah? Gonads. Parietal and visceral mesoderms line the intraembryonic xylem. Will line the intraembryonic xylem. Parietal mesoderm from the lateral and ventral body wall. Okay. Whereas the visceral mesoderm from the wall of the gut. Here's your future gut. Okay. So the visceral mesoderm from the wall of the gut, known as a serous membrane. So blood and blood vessels, beginning of the 3rd week, visceral mesoderm of wall of yolk sac differentiate into blood cells and blood vessels. So blood starting from the wall of yolk sac. Okay. So basically from mesenchymal cells, it's a blood island, forming the endothelial cells, primitive blood cells, and it's a luminal primitive blood vessels. On the wall of the yolk sac, this is a yolk sac, wall of yolk sac. They're forming the blood island, forming the blood vessels. Okay. How about the mesodermal germ layer? So basically we develop into supporting tissues, your connective tissues, all the muscles, striated and smooth muscles, blood, lymph cells, and wall of heart, blood vessel and lymph vessels. Okay. Kidneys, gonad, and corresponding duct, cortex of adrenal gland, spleen. Right? How about the endodermal germ layer? They are forming the gastrointestinal tract, the lining of gastrointestinal tract, the yellow color here. Now, how from the flat embryo, okay, the 3 layers, ectoderm, mesoderm, and endoderm suddenly become embryo-like structures. For example, this one. Can you imagine from these structures, a very flat, and then suddenly become a fetus-like appearance because of the folding. There are 2 foldings. Folding in bahasa we call it lipatan, folding, lipatan. So there are 2 foldings, cephalocaudal folding and also lateral folding. Cephalocaudal means head to the tail, folding. So they are something like folded. For example, the head is a tail, they are folded. And then at the same time, there's a lateral folding. Lateral folding will be this one, the lateral folding. It's a head and tail, they are folded. At the same time, lateral folding as well. Okay, so there are two folding. So who induced this folding? What structure causes this folding? So the cephalocaudal folding due to rapid longitudinal growth of C and S. So if you look here from the lateral view, the diagram here from the lateral view, the blue color here, because it's a lateral view, it's a developing CNS. Okay, because of the rapid development, it causes cephalocaudal folding. Cephalocaudal folding. And then the lateral folding, we are view from the anterior view here. All right. Lateral folding due to the rapidly growing somite. Remember the somite just now? On the paraxial mesoderm, we develop into somite. So these two structures causes the folding, then eventually develop into a proper fetus. So the yellow color here representing structures derived from the endoderm. So external appearance during the second month. Okay. End of week 4 of 28 somite, the main external feature are the somite and subpharyngeal arches. Age expressed in somite. So during second month, count ramen length CRL in millimeter. So CRL is a measurement of the length of human embryo and fetus from the top of the head count to the bottom of the buttocks. It is typically determined from ultrasound imagery and can be used to estimate the gestation age. Yeah? So this, for example, here, CRL from the top to the tail. Right? So during the second month, changed greatly by the enormous size of head. This is your head. Formations of the limb, upper limb, lower limb. Okay. The face, the ears, nose, and eyes. And beginning week 5, forelimb and high limbs are appears. Yeah? Okay, what's the teratology? The last topic, teratology. So basically, by looking at these pictures on the right side, showing the period of susceptibility to the teratogenesis. Yeah? So, from fertilizations to week 3 or week 2, not sensitive usually, okay, but high rate of mortality may occur. All right, but during the embryogenesis, from week 3 to week 8, all right, each organ system will also have a period of peak sensitivity. Means that they are very sensitive with the external factors or from the maternal factors. If the mother taking drugs or having a very dangerous lifestyle and so on. Right? By looking at this graph here, risk of formation occur during organogenesis or embryogenesis from week 3 to week 8. High risk. Right? Then after that, once the organ is already developed, then the risk will become lower. All right. So what is a teratology? The science that studied the cause of structural, functional, behavioral, and metabolic disorder present at birth. So 21% of infant death is due to birth defects. So major structural anomalies occur in 22% of life-born infant. Additional to 3 are recognized in children by age of 5 years. This teratology. So I think you can read these things very straightforward. All right. Among the birth defects, up to 60% is unknown causes. About 50% is genetic factors. 10% is environmental factors. up to 25% due to multi-factors, but 1% because of twinning. So it's the malformations, type of anomaly, malformation, disruption, deformity, syndrome. So you can read on your own. Okay, two major group, environmental and also genetic. Environmental can be physical, chemical, nutritional, hormonal, or maternal infections. This is radiation effects and so on. Right? This is a chemical agent like thalidomide, aminoterine, and so on. So these examples of the malformations. A baby without a limb, upper limb and lower limbs. Or it can be cleft lips, or it can be cleft lips and also cleft palates. All right. Okay. These are nutritional factors, hormonal factors. These are maternal infections, genetic factors. All right. So periods of susceptibility to teratogenesis. Okay. 0 to 2 weeks. First day of gestation is usually immune to teratogens. But in week 3 to week 8, period of greatest sensitivity. From week 9 to 38 is a decreasing sensitivity. It's a period of functional maturation.